This is my internship at the Noah Milford Lab. I'm Amber Gruno. I'll be attending UConn in the fall, Avery Point. I'll be majoring in marine sciences. My internship site was the Noah Milford Lab. They basically research shellfish, a lot of aquaculture things. They grow scallops, mussels, oysters, and they collect surf clams from different sites around Connecticut and the East Coast. They focus a lot in aquaculture, and they provide services for a lot of farmers around Connecticut. My supervisor was Lisa Milkey. She is the head of the aquaculture division at NOAA. And, sorry. She's been working there for over 18 years. She was my supervisor, but I was working with a lot of other people at the site as well every day. I chose this because I really love getting my hands dirty. I've only had experience working with finfish at Bridgeport Aqua, working in the hatchery. I really like fish husbandry, and I wanted to get into more of the research side, so that's why I picked NOAA. And yeah, I wanted to just learn a lot of different things and get a bit more experience. So basically they do a lot of, they culture a lot of their own specimens to research. They culture scallops in the basement, as well as the algae that they eat and stuff. Sorry. They research a lot of oysters and clams. Surf clams are what they get from different sites. They have a bacteria lab in the facility that they work on. Yeah. I didn't really know what to expect going into it. What I didn't expect also was to have another intern from Foreign working with me. So when I say we, that's what I'm referring to. Her name was Emma. She was really nice. I didn't expect. I was expecting to work on a lot of different projects. I was told to be testing shell strength, dissecting shellfish. I didn't really know exactly what to expect. I expected some husbandry, some maintenance, which I was used to working the hatchery. But other than that, I didn't really know. My responsibilities. So lots of cleaning, so much tedious work. Those are flasks that I cleaned in like a few hours. It was a lot. Labeling tubes is something I did a lot. It got pretty fast after like the 100th tube. It was a lot. There's the tank farm that they have on the site. They raise a lot of oysters and stuff in there. I was told that they don't really take care of the stuff in the tank farm, that you just kind of put it in there and whatever grows, grows, and they're kind of surprised when they check on it and there's stuff in there. Another thing I had to do one time was they had like a bin of chemicals that were just completely expired. Like there was one from like 1960 in there, and I had to document all the chemical names, the company, and the date it was opened, which is usually in the 1900s. It was crazy. There's so much old stuff there. I think the oldest thing I found in the building was from like 1906 in the library. It was crazy. Lots of dissections as well. We worked on the scallop project. The scallop project was ocean acidification study that they've been doing. It's like the overarching project in the facility. Basically, ocean acidification is carbon dioxide dissolves really readily into the ocean, so all of the CO2 emissions that are from the land get dissolved into the ocean. Carbon dioxide, it combines with oxygen to create hydrogen ions, which lowers the pH of the water and makes it much more acidic. And that takes away carbon that could be used, or calcium that could be used by shellfish to build their shells. So they overall just have less growth. So this is what this study is. With the scallops, it's like a multi-generational study of scallops. They started with one that has low OA conditions, moderate, and then high. And then from that generation, those get lower generations. So the low OA goes to low, medium, high. The moderate is low, medium, high, and they kind of just raise them through these different conditions throughout their lifetime and their generations and see how it affects their growth. The orange one here is the moderate OA, and then that one is high, that one's low. And we were basically taking the tissue out of one side of the shell, putting it into the other, and then moving around the tissue so that you could see the gonads, which are the reproductive organs, so that we could see if they're ready to spawn to make the next generation of the study. I learned a lot about shellfish anatomy here as well. Like, it's a little cartoon, it's really simplified, but this is the mantle right here. This is the gut, and the gills are under here a little bit. The gonad is going to be bright orange if it's a female, and then if it's a male, it'll be really dark black with like a white stripe going down. And the whiter the stripe, the more mature it is, and the more bright orange it is, the more mature it is. I also detected a lot of surf clams that they collected from sites around Massachusetts. I learned a lot about clam anatomy as well. Like, that one's super simplified as well, but there's a gut, the mantle, the same here, and it's like a big foot coming out. They're really cool. But we were measuring their length, height, and thickness, and putting it in a data sheet, and then just taking all the tissue out of the clam and putting it into a separate tray, and labeling where the shell is so they go together. And they were like weighing it to see how much mass is in that tissue, basically. That project was for seeing how they are doing in the wild with their growth, basically. And yeah. This was more of the scallops. This necklace I'm wearing is actually one of the scallops that we dissected. Oh, cool. Yeah, that was really cool. You could see, on average, these guys are much bigger than those guys are. I think this entire group is a generation that came off of the low OA conditions. So, yeah, you can see that it very, very much impeded their growth. Something else we did with the scallops, one of the researchers had their own little like personal project they wanted to do along with the scallop project. They wanted to see if ocean acidification conditions can affect the coloration of the scallop shells. So we were struggling to differentiate like light brown, brown, tan, like brown with white or like white with brown, and it was it was a lot, and definitely got a little bit better at making data sheets, like differentiating different conditions and things like that. So that was rough. I also worked with mussels that they were growing there. Those were for a study trying to create different strains of mussels that could be more resistant to different conditions in like aquaculture farms, so they could like sell these mussels to, or not sell them, but like I think they were selling them, but to different farms to like better survive against like changes in temperature or dissolved oxygen, different conditions like that. We were helping them spawn the mussels, sorry. And whenever we watch with a flashlight to see if they started like making sperm or eggs, and if we saw that, then we would isolate them in these big trays, and it got a little chaotic. There was a lot more than this at the end. We were just stacking trays, and then eventually they collected them at the end and then mixed everything around, and we got a lot of baby mussels, like maybe like 1 million baby mussels, but then eventually like a lot of them won't survive, so there'll be like a few thousand at the end. But that was really fun, a lot of chaos. Another project that someone was working on there that we helped with, they were collecting samples of oysters across, I think it was the East Coast, yeah. They were just going up and down and comparing the genetics of wild oysters to oysters raised in farms and seeing if there's any correlation, any differences. I also learned a lot about sanitation here. When they were shucking the oysters, which is like opening them, and then taking tissue samples and putting it in little tubes of ethanol to preserve them for a while so that they could analyze them later. And they would go down this line of, I think it was a freshwater bath, and then a bleach and like sand bath, putting their tools in, and then salt water, I think or alcohol, and then they would flame it over flame to completely disinfect it. So it's like a really long process of being very thorough with this. There is one of the oysters that they found, it had a really bad boring sponge problem. You can see the top of the shell is like super porous, and the way it works is boring sponges like attach to the top of the shell and eventually just kind of burrow into it and destroy it from the inside. This is a really bad one. It's all orange on the inside. This is a picture of oyster gonads. The dark purple is sperm, and then the lighter pink is going to be eggs. The darker it is the more mature it is. So this one was really dark and that one was really light. That was really cool to see under the microscope. I also got to go on the research vessel, the Victor Lucenoff. It was really fun. Basically we went around Charles Island, and checked on their oyster cages that they had set up. They had like 6 of them. We were putting cameras on them to try to monitor how fish interacted with these. I think it would take about like 8 minutes of data every hour, at the beginning of every hour. And then they would analyze all these clips and see how they're interacting with these basically artificial reefs. There was a lot of fish around them. When they pulled them up, there was like fish flopping off of the cages. It was really cool. So that was a really cool experience. This is the tank farm. I helped to clean one of these tanks. We like took these big, long squeegee mops and was just like sweeping the bottom of it after we drained it. That was really cool. They have these oyster cages where they grow oysters for another project that some contractors are doing there that I was helping with. They were growing oysters to be, again, more resistant to different conditions in aquaculture settings. And this is their upweller systems that they had, these like bottles, glass bottles. They were a new kind of farming oysters. I think the larval stages would go in there. I think it's the first facility in the US that's using this method right now. That was really fun to work on. I also helped with raising the algae that they grow on the site. They have media up here that feeds into these algae flasks that helps them grow a little bit, so they kind of like harvest half of it and then it just grows back over time. So they harvest it like every week. This is a continuous thing. But because of the scallop project, they need like mass amounts of algae for this project. So they got these massive automated algae machines, or they have like gallons and gallons of algae in there, and they just press some buttons on that little screen, and a tube like feeds out some algae into a bucket, and they just take it to the shellfish, feed them. So it's just a much more like mass-produced way of making algae, whereas this is just for like the smaller tanks. I also worked with the microbiologist on the site, Diane. She was really cool. This is us making marine agar for bacteria to grow on. We heat it up and mix it around and stuff. There's like a little thing, little mixing, I don't know what to call it, but when it was heated up, it would like spin around and mix it for us, which was cool to see. We would pour these different medias into little trays. I think this is like a tryptic soy agar, which is just an all-purpose media for different bacteria to grow on. The middle one is marine agar, which is a lot of different nutrients that's in seawater, and that would isolate bacteria that like to grow on that. And then TCBS is the green one. That would isolate Vibrio that grow in seawater. They're not really harmful. They're just kind of bacteria that grow around in seawater. We would also put some of this marine agar in little tubes and make it grow or make it solidify at a slant, so that we could more easily put the little loop in there and streak bacteria from it instead of having to like awkwardly go in there. This is Diane flaming the loop to sterilize it before she tries to take some of the bacteria that's growing on the agar and put it into there. This is how the bacteria grows on the agar. It kind of makes a streaking pattern. And that's us after, some of the scallops in the hatchery started to die a little bit. So Diane wanted to test if it was going to be a problem with the algae that they're eating. So she took a sample of it and put it over some of the different media plates and saw what grew, basically. So that's us helping to put samples of the algae on the plates. That was really fun. Another thing, she gets commissions from different farms around Connecticut as well, or just around the area. And one of them was an oyster farm who all of their oyster larvae died. So they wanted to figure out if it was in the water, if it was like a bacteria growing. So they sent her a sample of the seawater that they were using, and she was trying to figure out what it was. She put it on marine agar. It seemed to really like that. It was growing a lot, but it was also starting to turn black, which could be a very, very dangerous bacteria for shellfish. So I wanted to test if it was a harmful bacteria or something that was just a Vibrio. She did something called a gram stain, where we applied different dyes to a sample of this bacteria and let the cell walls absorb it into the cell, and we would then wash away the dye, and if the cell walls were strong, then it would retain the dye and stay a dark purple, but if the cell walls were weaker, more thin, they would expel the dye and it would be like a lighter pink color. That the lighter pink color would be gram-negative and the stronger one would be gram-positive. Gram-negative is what we wanted, and that's what we got, thank God, because apparently we would have a big problem. That was cool. Some challenges I faced, finding things to do, especially towards the end, got really hard. Things got a little bit chaotic and there was like less places for me to help out during or in. So at some point I was just shredding papers, helping people organize things, just cleaning. That's where I like did all those dishes and didn't really have too much to do at the end, but it was still really fun. Asking questions was something I struggled with at the beginning a lot. I definitely learned how to ask more questions towards the end. Another thing I struggled with, just the diversity of the tasks that I was given. Like one day I was working with the scallops and the next like hour I was working with Anne in the lab doing like bacteria things, and it was just a little bit difficult to switch from one thing to the other and still like remember everything that I did. So that was a bit hard. I definitely have a really solid foundation of a lot of different things now. Like I had never dissected anything before this, but now I've done like hours of dissecting, and that was really cool. I learned different like harder skills like shucking oysters. I didn't do it myself, but I learned the process to like not pierce the gut and like ruin your sample. Dissecting was one thing. Sanitization, I didn't realize it was that intense in a research setting, like just how much you have to make sure everything is sterile. Just shellfish culture in general, like the process of raising larval shellfish, they're like microscopic. I learned that when they're like microscopic like that, they need something to set onto so they can start growing. So you fill their tank with just grains of sand, and they're so tiny that they settle on the grains of sand. So every oyster you'll find like just out in the wild will have a little tiny grain of sand on their hinge, because that's what they started on, which is just really cool. I learned a lot about shellfish anatomy through all the dissections that I did. They're really cool. For my future, all that knowledge that I gained, I definitely know a lot more about the industry. I was very focused on fish husbandry before and going into college for fish husbandry, because that's something I just really enjoy, but I never really had an experience with anything else, like shellfish. I had no idea about anything about shellfish before I went into this. And now I also met a lot of people there who graduated from Avery Point, which is a really cool sign. They told me about a lot of professors that were cool there, a lot of people they knew, told me some major advice, and now I was kind of like set between like, oh, I kind of really want to go into aquaculture, but I also kind of like research as well. But this showed me that you can combine them as well and research aquaculture, which is always going to be something that's growing, so it's kind of new as well. So it's definitely something that I think I could go into in the future. I also intend to participate in a work study at UConn, so I think this is really going to help me choose a topic and be able to have all these skills that I can use for my research. And that's it. Got any questions?
Yes. Amber, I'm so impressed. Like the sheer amount of, like you were just saying, the knowledge you gained, like the experience you had. Do you feel like anything at your aquaculture experience here like prepared you for what you were going to do, or did you feel like this was an extension of some of the things you already knew?
I think, yeah, a little bit of both, extension, and I had a really good foundation in aqua. Like, I was really used to cleaning tanks and maintenance like that. Yeah, I knew like a lot of the like parts of the system. Like I knew where the pump was. I could point out like the temperature. Oh my god, I'm blanking on the word, but what controls the temperature. Like the sump, I knew all the parts of the system. I didn't really know anything about shellfish, but we didn't really focus on shellfish at all at Aqua, actually. Pretty much just finfish. And like, yeah.
Do you have an affinity still for finfish, or would you think you'd actually like to research more about shellfish? Do you have like an affinity for one over the other anymore?
I don't really think so. I think I'm kind of like,
Like both?
yeah, I like them both.
I think it's really interesting at the end when you're talking about the future, you're kind of like talking about like research or exploration, correction, Agriculture,
Yeah.
but like the research side, or again,
Yeah.
you were kind of torn on like what to do, and it sounds like this experience opened your eyes a little bit to say like it could be both. Like you don't
Definitely.
have to focus on one or the other. So I think for you, I'm so thrilled it worked out for you because it sounds, I know you said at the end it got a little like dull, but it sounds like in the time you were there, you like going out on the boat, I think it's fascinating
Yeah.
to be able to see like, because one of the questions I had as you were like starting to present was where did they obtain all of these like mussels and scallops and oysters, and I know there's like oyster farms that you can go out to, so
Yeah.
does Noah have their own like farms, or do they get it from like, lack of a better term, like do they like sub out like the
Samples?
They a little bit of both. They do have like I remember going to they have like a basement level that basically grows all the shellfish. They have trays of mussels that they're growing right now, and recently oysters. They have many trays of scallops, so many scallops. The tank farm also grows scallops and oysters, I believe. I think clams. I'm not really sure about that. But they also have a lot of sites they go out to, and the oyster cages they keep as well around Charles Island. Yeah, they have a lot of sites to go out to and collect oysters and clams from as well.
So cool.
Do you eat, okay, I have very random questions for you, but do you eat shellfish?
I don't think I actually tried shellfish until this internship. So, funny thing, at one point they had like a pizza party at the site one time, and they had clam pizza there. That morning I had been dissecting clams all morning, and I had that smell of like dead shellfish in my face. I didn't realize it was clam pizza. So I took one. I was like, this tastes like what I was doing this morning.
Did you like it?
It was good. I think if I hadn't been doing that morning, I would have liked it more. But yeah.
Yeah.
That was a good story.
And now, are there any opportunities for you to continue like either volunteering there or a job, or like will you keep in contact with your site supervisor going forward?
I think I will. I think they do have scallop spawning is what they mentioned the first time I got there. It's a really chaotic process apparently because scallops are hermaphrodites, meaning after they spawn they change gender. So like if one produces eggs and then like suddenly they're producing sperm and they could fertilize their own eggs, which is like really dangerous. So it's like everyone is working on this, and it got a little bit postponed because of the problems with the scallops. But I might be going back to help out with that soon.
Oh, cool.
So
Awesome.
Yeah.
Such a great resume builder. We're so proud of you. You did such a great job, and what a good representation of law at such a cool like facility